A warm welcome to one and all. Today, dear friends, we are here to attend to learn the topic endoplasmic reticulum and the structure and functions of endoplasmic reticulum. This is the first unit of semester second and the last topic of first unit. So, let's start today the endoplasmic reticulum, its ultrastructure and functions. So, first introduction of endoplasmic reticulum. In the year 1945, the lace-like membranes of the endoplasmic reticulum were first seen in the cytoplasm of chick embryo cells. These are membrane-bound channels seen in the form of a network of delicate strands and vesicles in the cytoplasm. These are single membrane cell organelles. These form an interconnected network of tubules, vesicles and cisternae within the cells. Endoplasmic reticulum are considered as one of the components of cytoskeleton. As I have explained before that what is cytoskeleton? The skeletal support which is given to the cell is this endoplasmic reticulum. So, it is called as cytoskeleton which is present in cell cytoplasm. So, this is called as cytoskeleton along with microtubules, microfilaments and intermediate filaments. These are all muscle filaments which gives the skeletal support to the living cells. But in a single living cell, the skeletal support is given by this endoplasmic reticulum. These are first of all observed by Porter, Claude and Fullman in 1945 as a network. The term endoplasmic reticulum was first used by Porter and Fullman in 1952. See friends, the structure was first observed by Porter, Claude and Fullman in the year 1945. From 1945, after the 7 years, that is in 1952, Porter and Fullman give the structure the name, the endoplasmic reticulum. So, this is all about the introduction of endoplasmic reticulum. Now, where it is located? In a living cell, where the endoplasmic reticulum is located. So, it is present in almost all eukaryotic cells. These are found to be absent in mature erythrocytes, ova, embryonic cells and prokaryotes. So, this is a question which is asked many times in the competitive exams or in MCQ test, that where the endoplasmic reticulum is absent in which cells. So, in the mature erythrocytes, ova, embryonic cells and prokaryotes it is absent. The endoplasmic reticulum often occupies most of the cytoplasm. Now, amount. How much amount of endoplasmic reticulum is present in which cells? So, let us see. The ER varies in amount from cell to cell. In spermatocytes, it is represented by a few vacuoles only. In mammalian spermatocytes, mature sperms, the amount of endoplasmic reticulum is very less. It is found in the form of vacuoles only. In the cells of adipose tissue, it is quite simple, having the form of few tubules. Only few tubules are present in adipose tissue. The cells that are actively synthesizing proteins such as liver cells and pancreatic cells and fibroblasts have abundant endoplasmic reticulum. So, see, very rarely it is seen in spermatocytes. Then in adipose tissue, some vacuoles are present in vacuole structures or tubule structures are present, which are the structures of endoplasmic reticulum. In ample amount, it is found in the cells which synthesizes proteins. Now, which cells synthesize proteins? So, liver cells, pancreas cells and fibroblast cells. In these cells, the endoplasmic reticulum is found in ample amount. Endoplasmic reticulum forms 30 to 60% of the total membrane in the cell. Now, origin of endoplasmic reticulum. At present manner of origin of the endoplasmic, it is not definitely known. Till now, the definite origin is not known. The most concrete hypothesis is that the ER is budded off from the nuclear envelope which was invented or discovered, or this statement is given by Wichneter Nitzer in 1974. So, this is the theory which was produced by Wichneter in 1974, that ER is budded off from the nuclear envelope itself. That means it was the part of nuclear envelope. So, the ER appears to arise from the outer membrane of the nuclear envelope by outfolding, or from the plasma membrane by infolding. The smooth ER seems to arise from the rough ER by detachment of ribosomes. So, this is all about the origin. There are two basic morphological types of ER, namely RER and SER. RER stands for rough endoplasmic reticulum and SER for smooth endoplasmic reticulum. The endoplasmic reticulum ER membrane is thinner, that is 50 angstrom, than that of plasma membrane. The thickness of plasma membrane is 80 to 100 angstrom, and of ER is 50 angstrom. See the structure here, it is the ultrastructure, electron microscopic structure of endoplasmic reticulum. So, this is smooth endoplasmic reticulum. These smooth surfaces of bag-like structures or cisternae or tubules, it is smooth ER. These cisternae or space, these are rough endoplasmic reticulum. It is called as rough ER because of the presence of or attachments of these ribosomes on its outermost surface. These are all cisternae. This blue or purple color is of nuclear envelope, and these are the nuclear pores which are present in the nuclear membrane. Now, physical structure of ER. The ER is three-dimensional network of intracellular. It is formed of three types of elements. Three types of structures togetherly forms the structure of ER. The first is cisternae, second is tubules, and third is vesicles. Now, number one, cisternae. These are flattened, unbranched, sac-like elements. They lie in stacks parallel to one another. They bear ribosomes on the surface and therefore appears rough. It contains glycoproteins named ribofourin 1 and ribofourin 2 that binds the ribosomes. These two proteins, namely ribofourin 1 and ribofourin 2, are responsible for the attachment of ribosomes on the outermost surface of rough ER. So, please understand these two names, these are important, which are responsible for the attachment of ribosomes. One is ribofourin 1 and another is ribofourin 2. Now, the second structure is tubules. These are irregular branching element which form a network along with other elements. These are often free of ribosomes. Tubules are free of ribosomes. That it means that cisternae consists of ribosomes, tubules shows absence of ribosomes. Now, the third structure is vesicles. These are oval and rounded vacuole-like elements. These are also free of ribosomes. So, dear friends, ribosomes are completely absent on tubules and vesicles, but only cisternae shows presence of ribosomes. All the elements of ER freely communicates with one another and contains a fluid called endoplasmic matrix in the ER lumen. This matrix is different from cytoplasmic matrix outside the ER. The ER may pass from one cell to another through the plasmodesmata in the form of desmotubules. Now, again the structure here, this is the central nucleus. Just you imagine, friends, that this is the complete cell structure, living cell structure, and in this cell structure, this white color is cytoplasm. This centralized structure is nucleus, which is showing the presence of nuclear envelope. In the nuclear envelope, small pores are present, which are called as nuclear pore. Now, these are smooth endoplasmic reticulum, which shows absence of ribosomes on its outermost surface, and these are the bag-like or stack-like structures or cisternae which shows the presence of ribosomes on its outermost surface. Hence, these are rough endoplasmic reticulum, these are ribosomes, and these are cisternae. The space between the two cisternae is called as cisternal space. Now, molecular structure of endoplasmic reticulum. The membrane of ER are composed of two layers of phospholipids. Dear friends, as in the first topic, I have explained you the structure of plasma membrane. It is double layered, double phospholipid layer. So, that structure is found here. The membrane of ER are composed of two layers of phospholipids molecules sandwiched by two layers of protein molecules like other membrane in the cell wall. The types of ER: smooth ER and rough ER, as I have explained previously in the previous slide, SER and RER. Now, endoplasmic matrix. This is the matrix. The space inside the tubules and vesicles is filled with a watery medium that is different from the fluid in the cytosol outside the ER. It is called as endoplasmic matrix. Their walls are constructed of lipid bilayers, membranes that contains large amount of proteins similar to the cell membrane. Now, smooth ER. From the two types of ER, the first one is smooth ER. Smooth ER is an arrangement of tubules, vesicles and sacs. The size and structure of SER varies between the cells. The SER can change within a cell's lifetime to allow the cell to adapt to changes in its function and requirements. As per the requirements and its functional capacity, the structure or cell size can be changed. So, this type of function is seen in smooth endoplasmic reticulum. There are no ribosomes attached to the membrane surface. The SER is connected to the nuclear envelope. The network of the SER allows there to be enough surface area for the action or storage of key enzymes or the products of the enzymes. The SER is less stable. The SER is characteristics of cells in which synthesis of non-protein substances takes place. The function of SER is a characteristics of cells in which synthesis of non-protein substances takes place. Okay. Now, the second type is rough endoplasmic reticulum, RER. The surface of the RER is studded with ribosomes, giving it a rough appearance. It mainly consists of cisternae. The membrane of RER forms large double membrane sheets, which is located near and continuous with the outer layer of nuclear envelope. Rough ER is very important in the synthesis and packaging of proteins. Example: Russell's bodies of plasma, Nissel's granules of nerve cells. So, in plasma, that is in blood, and in the nerve cells, the protein is synthesized, and this protein is synthesized due to the presence of RER, rough endoplasmic reticulum, and the ribosomes present on its outermost surface. These organs are responsible for this protein synthesis and packaging. So, binding site of the ribosome on the RER is the translocon. The ribosomes bound to the RER at any one time are not a stable part of this organelle's structure, because ribosomes are constantly being bound and released from the membranes. As you have seen in the structure that the ribosomes are bounded on the outermost surface of the rough ER due to the presence of due to the presence of two proteins, the riboflavin 1 and riboflavin 2. So, these two proteins are responsible for the binding of ribosomes. So, when this detaches, these ribosomes detaches also and bounds also. This process is constantly going on. So, it is like a cycle that the ribosomes get attached or bounds at the outermost membrane and get removed from it and moves freely in the cell cytoplasm. The ribosomes only binds to the RER once a specific protein nucleic acid complex forms in the cytosol. This special complex forms when a free ribosome begins translating the mRNA of a protein destined for the secretory pathway. This is the process in which protein synthesis takes place when mRNA from the nucleus or cell cytoplasm comes in contact with ribosomes. Then these two, that means mRNA and ribosome, along with transfer RNA, tRNA, secretes the protein or synthesizes the protein. The first 5 to 30 amino acid polymerized encode a single peptide, a molecular message that is recognized and bound by a single recognition particle, SRP. Single recognition particle is SRP. Now, the ribosomes that become attached to the endoplasmic reticulum synthesizes all transmembrane proteins. The most secreted proteins that are sorted in the Golgi apparatus, lysosomes and endosomes. Translation pauses and the ribosomes complex binds to the RER translocon. Now, protein transport. As proteins are formed in the endoplasmic reticulum, they are transported through the tubules towards proteins of the SER, smooth endoplasmic reticulum, that lie nearest to Golgi apparatus. At this point, small transport vesicles composed of small envelopes of smooth ER continually break away and diffuse to the deepest layer of Golgi apparatus. Inside these vesicles are the synthesized proteins and other products from the ER present. See the process here, as we have seen, as I have told in the previous slide. This is the nuclear membrane. This is the presence of rough endoplasmic reticulum. From these cisternae or these tubules, these vesicles are generated. These green color vesicles are generated. So, this is the vesicle, this is second, this is third vesicle. These vesicles contains the protein material or other elements which are present in these cisternae. So, that element get transported through these vesicles from rough ER to Golgi apparatus. This is the cell membrane. This is the cell membrane of Golgi apparatus. This is the Golgi apparatus and this is the cell membrane. That means cell wall of the living cell. So, the transportation takes place. This is the Golgi apparatus where all these proteins are stored, and at the time of requirement the proteins are transported again from Golgi apparatus to the cell membrane. So, this is all about endoplasmic reticulum. Second method of transport out of the endoplasmic reticulum involves areas called membrane contact sites, where membranes of the endoplasmic reticulum and other organelles are held together, allowing the transfer of lipid and other small molecules. Now, the types and functions of ER: rough endoplasmic reticulum RER, smooth endoplasmic reticulum SER. Let's see. Functions of RER: surface for ribosomes. So, rough endoplasmic reticulum provides a surface for the ribosomes to attach. The RER provides space and ribophorins for the attachment of riboforin 1 and riboforin 2 for the attachment of ribosomes to itself. Surface for protein synthesis takes place: formation of glycoprotein, synthesis of precursors, then smooth endoplasmic reticulum formation. The rough ER gives rise to the smooth ER by loss of ribosomes. As the ribosomes from the outermost surface of RER get removed, it get converted into smooth ER. And functions of SER, that is smooth endoplasmic reticulum. The smooth endoplasmic reticulum lacks ribosomes and functions in lipid metabolism, carbohydrate metabolism and detoxification, and is especially abundant in mammalian liver and gonad cells. So, these functions in lipid metabolism, carbohydrate metabolism and detoxification, these are the functions of smooth ER. It also synthesizes phospholipids. Phospholipids means the lipids which forms the plasma membrane or cell wall of the cell. So, these phospholipids are synthesized in these smooth endoplasmic reticulum. Cells which secrete these products, such as those in the testes, ovaries and skin oil glands, have a great deal of smooth endoplasmic reticulum. Detoxification. The SER brings about detoxification in the liver, that is, converts harmful materials, drugs and poisons into harmless ones for excretion by the cell. This important function of detoxification is performed by smooth endoplasmic reticulum. Formation of organelles. The SER produces Golgi apparatus, lysosomes and vacuoles. These all organelles are formed by the smooth endoplasmic reticulum. It also carries out the attachment of receptors or cell membrane proteins and steroid metabolism. In muscle cells, it regulates calcium ion concentration. It functions as calcium ion concentration and regulates the calcium ion concentration in the muscle cells. The smooth ER also contains the enzyme glucose 6-phosphate, which converts glucose 6-phosphate to glucose, a step in gluconeogenesis. So, in carbohydrate metabolism, it functions in this way. Now, dear friends, this is the link. I don't know it will start or not, but no. So, this is all about the endoplasmic reticulum, its type, its ultrastructure, its functions. So I have created this lecture for the two topic togetherly, as the ultrastructure of ribosomes and functions of ribosomes. So dear friends, I think you all understood it very well, and if there is any query or any difficulty, please ask me in our WhatsApp group. Thank you once again.